he said that in taxonomy, some portion also completed. So anyway, so I will start with the anatomy part. After complete anatomy, next to taxonomy, if you have any time, if you have any enough time, again I will complete gymnosperms also, right? And you don't worry, already gymnosperms, anatomy, taxonomy, you must have learnt in during the practical training classes. Very recently completed those training classes also. So I think no problem with C3, taxonomy part and gymnosperms anatomy, right? So let me start with anatomy part in this. First of all, what is anatomy? What we study in anatomy, plant anatomy. Hello?
Internal structure, sir.
What are the internal parts?
Xylem, phloem.
Right, xylem, phloem. Only two present, xylem and phloem.
Pericycle.
Okay. How they are formed first? How they made up of? Xylem, how made up of xylem and phloem? What you said, pericycle. So the part is group of.
Meristematic tissue.
Okay, internal meristematic also present. First of all, what is tissue?
A group of same cells.
Group of cells. The group of cells called as tissue, right? How many types of tissue in plants?
Two,
Two
sir.
types.
What are those?
Permanent tissue.
Right.
Meristematic.
Permanent tissue and meristematic tissue. Right. So let me share PPT here. Are you able to see the tissue slide?
Yes, sir.
Right. As you said here, the tissue is the group of the cells. What is cell?
Structural functional unit.
Why it is structural and why it is functional? Structural means it is forms the body of an organism, body of the plant. Right, the body which is with the help of the permanent tissue, which may be parenchyma, collenchyma, sclerenchyma, xylem, phloem, they are all constituted in the formation of the part of structural units, structural parts. And if you take any functional unit, like photosynthesis, respiration, whatever it may be, these are all functional unit takes place inside the cell. That's why it is called as functional unit. Right. So anyway, if you take the plant tissues are classified into two major permanent and meristematic tissue. What is different between permanent and meristematic tissue? First of all, how they form in embryonic. First, meristematic tissue, meristematic cells formed from the embryonic cells. This embryo. These are the embryonic cells, for example, embryonic cell. This embryonic cell divided into two cells. Meristematic cells, mainly from the embryonic cell, meristematic cells are formed. M for meristematic. These meristematic cells divide mitotically, redivide again. That means continuously divide and forms one one meristematic cell, one permanent cell. Again, meristematic cell, M for meristematic. Divide and formation as a meristematic and permanent. But what you observe in this, always which cell divide? Always which cell divide in this? Meristematic cell or plant permanent cell?
Meristematic cell.
Right, meristematic cell divide and redivide. And in this division, the always forms one cell meristematic, another cell permanent. If you observe the permanent cell cannot divide temporarily, but not permanently. Of course, whenever it is necessary, the permanent cell also can metamorphose into meristematic cell, can metamorphose into meristematic cell and perform as a meristematic cell. Okay. Then what happened during this division from embryonic cell? From embryonic cell, all time division redivisions occur in the part meristematic cells, group of cells formed and permanent group of cells formed. Like that, here the meristematic tissue formed and also permanent tissue formed. But which portion majority in this meristematic cell or permanent tissue? Meristematic tissue majority or permanent tissue majority?
Permanent tissue.
Permanent tissue, because here the permanent cell accumulated one for every division. But what happened in meristematic cell? But meristematic cell divide and redivide continuously. That's why for every division, always it is divided, but permanent tissue accumulated. That's why permanent tissue number increasing gradually. Right? Is it clear? The formation meristematic and permanent first of all.
Yes, sir.
Right. Already you know how the embryo formed. I don't go to in detail about how embryo formed, fertilization, male gamete, female gamete fusion. Okay. From embryo, after formation embryo, embryonic cell develops into meristematic. These meristematic cells divide, forms permanent cells like permanent tissue form. Okay, right. If you take, I think comparison somewhat clear. Meristematic between permanent. Meristematic, it is always divided, redivided. Permanent tissue not divided temporarily. Not, of course, whenever it necessary, the permanent also can divide. Remember that word also. If you take the meristematic types, in this apical meristem, intercalary meristem, lateral meristem. So this is based on position. I will clearly explain about meristematic tissue. This is long question, very, very important. I will explain later about meristematic tissue, right? And let's say permanent tissue. In permanent tissue, again, simple and complex. What is different in simple and complex? Simple, similar type of cells, function and origin, similar type. But in complex, different type of cells, function same, but structurally different type of group of cells are there in complex tissue. For example, see, if you take the simple tissue, parenchyma, one type, collenchyma, another type, sclerenchyma, three types. If you take parenchyma, the origin and function, the parenchyma same. If you take collenchyma, same origin and function, same in collenchyma, similar type of cells, the same similar type of cells in sclerenchyma. They are individual, parenchyma individual, collenchyma, but not together. So that's why it is individual, that is simple tissue. In complex, when the both together, that is complex. But function similar. Function similar means, for example, if you take xylem is the one of the complex tissue, phloem as well. But What is present inside the xylem? Different type of tissues. Already told you, simple similar type, complex different type. Let's see what are the different type in complex. Tracheids, trachid cell, vessel cell, xylem, xylem parenchyma, xylem fibers. If you take individually, tracheids one simple tissue, vessels one simple tissue, xylem parenchyma simple tissue, xylem fiber simple tissue, but all together that makes the complex and form as the xylem element. A similar way, phloem, sieve tube element, companion cells, phloem parenchyma, phloem fibers, they are all right. Is it clear? Hello?
Clear, sir.
Right,
Clear, sir.
right. Now let's discuss one by one. First, let me complete about meristematic tissue. Right, if you take the meristematic tissue, already given in our book, the meristematic tissues, some of the characteristics. What are the characteristics of meristematic tissue? These meristematic tissues very small in size. They are without intercellular space arrangement. They are inactive, always divide and redivide, and rich abundant mitochondria present when compared with permanent tissue. Abundant meristematic tissue, sorry, mitochondria is present inside the meristematic cells. They are the characters of meristematics. But here the important thing is a meristematic classification. Classification very, very important. So anyway, this is very, very important. Meristematic cells or types of meristematic tissue. Right. Are you able to see the slide? Classification of meristematic.
Yes,
Yes,
sir.
sir.
Right. Do you have book? Do you have book with you now at present?
Yes, I have.
Right. So here the first meristematic characteristics given, page number 350. But they are very simple, no need to explain about that. The simple, they are general, thin-walled, and without intercellular space, made up of cellulose, chemical cell wall made up of cellulose. And these are the just general characters, but not important in competitive or theory as well. But here the important is only page number 351, 16.4, classification of meristem. Right, this is classification. The meristem classification based on origin, position, plane of division. Three manner. First, based on origin, promeristem, primary meristem, secondary meristem. Based on position, in diagram given the based on position, apical meristem, intercalary meristem, lateral meristem. Next, plane of division, mass meristem, plate meristem, rib meristem. So based on function also actually not given, I think. Another one is right. Another type is based on function also, but not given in this slide. But remember, function I am right here. Based on function, got it. Already it has present in our book. Based on function, how many types given? Tell me based on function. See in book. Based on function.
3 types, sir.
What are those?
One is protoderm,
One is protoderm, like this outer protoderm.
second one is procambium,
Second one is
procambium,
procambium.
third one
Next
is
one, ground tissue.
ground
Next
merit.
one, this is right. Ground meristem, and it forms the ground tissue. Okay, right. Ground meristem. Right. So mainly the classification 4 types. Remember, first thing. Right. Let's discuss one by one. First, based on origin. Based on origin, promeristem. What is promeristem? Pro means. What is promeristem? Pro means.
First,
Right.
you.
Primordial meristem. That is embryonic meristem. Already told you in beginning. So the meristem where the foundation for new organs or their parts laid down, the referred to meristem or primordial meristem. The simple word embryonic meristem, embryonic cell. That is first. That is first form. From this promeristem, the second meristematic cells are present. That is primary meristem. Primary meristem. Right? It is the first derivative from the promeristem. Already in introduction, I have clearly explained embryonic cells. This embryonic E for embryonic, for example, embryonic cell. Promeristem, PM, promeristem, embryonic cell, development of promeristem. From promeristem, primary meristem will develop. Primary meristem develop. Right? So this is the first derivatives, the fundamental part of plants. And this meristematic persistent in the body part right from the embryonic stage. From beginning, already told you from embryo, from beginning, from embryonic cells, primary meristem, sorry, promeristem, next primary meristematic cells form. So these are from beginning, right? And this is involved for the development of body. And what you see in the position apical, intercalary, lateral, they are all development from the primary meristem. So this mainly the primary meristem mainly involved in development of the body. And also he has given clearly they are seen in stem apex, root apex, and primordial leaves, and the primary cambium, vascular bundles, and the intercalary meristem. They are derivatives from the primary meristem. These are all parts. And next, secondary meristem. Secondary meristem, actually, it is not present from the embryonic stage. In embryonic cell, the secondary meristem not present, which is development from second time. How it is developed from second time? For example, already told you beginning, primary meristematic cell function, division, redivision, involved in formation of body. But one meristematic cell divide, formation, one meristematic and permanent. Already told you many times. Permanent, right? And again meristematic divide and again formation permanent like this. But what happened? Whenever body necessary to formation the extra organs or extra parts, extra tissues, sometimes this permanent cell metamorphoses as a meristematic cell. This is called secondary meristematic cell. How the secondary meristematic tissue develop from embryonic or from permanent? Hello? How the secondary meristem develop? Hello?
Developed from the already developed tissue, secondary meristem.
Uh, uh, right, already developed. This already developed existence tissue developed from
Xylem is primary xylem and phloem. Secondary growth, sir. From
of course,
primary
of course.
xylem to metaxylem.
Okay, function of this secondary meristemic tissue, secondary growth involved in secondary growth. Okay, I will explain later that also. But what is the previous cell?
Primary meristem.
Right, primary meristem. Okay, right. Right, primary meristem. But of course, it is indirectly embryonic cell. Is it correct or not?
Yes, sir.
Right, right. That much I want. Clear about the origin?
Yes, sir.
So the secondary tissues involved in the secondary tissue formation like vascular cambium, cork tissue. I will explain clearly in secondary growth. But remember these words, whatever given in our book. So these are involved in cork cambium, intravascular cambium, and in both stem and root. Right. Now the based on position. In clearly we have seen in the diagram based on position, apical meristem, intercalary, lateral meristem. First of all, apical, where is apical meristem? Why, right. While the division takes place, some of the meristematic cells or tissue differentiated into some of the portion, deposited in some of the portion. Some migrate toward the apical, settled in apical meristem. Some are nodes and internodes, settled in intercalary. Some are in vascular bundle. They are lateral meristem, right? So first, let me complete about apical meristem. The name itself given apical. This apical meristem present at shoot and root apex. The function of apical meristem should be
Growth.
which
Longitudinal
growth?
Growth.
Right,
Apical
longitudinal growth, apical growth, or vertical growth, whatever it may be. They're all similar, right? And next, intercalary meristem. Where is intercalary meristem?
at the nodes region, sir. Nodes
At the
and
nodes
internodes.
region. These meristems occur between two permanent tissues. That means this are next region completely permanent. Either side permanent tissues are there, and derived from the apical meristem. This apical meristem already told you they differentiated meristematic tissue first apicals from the apical differentiated towards the nodes internodes. Now this settled as the intercalary, right? And this intercalary meristem present at the base of the leaf, many monocotyledons. And in grasses, these meristems play a role in vegetative reproduction. Already, what is the function of meristems? Regeneration, growth. That's why it is easily involved in vegetative reproduction, especially in grass family. And now lateral meristem. This is lateral. This is lateral meristem, which is present inside the vascular bundle. To take, for example, this is vascular bundle. In vascular bundle, for dividing xylem and phloem, let me change color. Meristematic tissue present between xylem and phloem, blue color. This is called lateral meristematic tissue. This is called lateral meristematic tissue. Right, let's see. Alongside the stem, parallel to the surface, Example, vascular cambium, gymnosperms and angiosperms. This is vascular cambium. Vascular bundle, vascular cambium, this one. Right? As it is positioned at the lateral side, which is called as lateral meristem. Right? And what is the function of lateral meristem? Hello?
It forms the plant body.
And it is also involved in secondary growth. The first of all, primary, the apical and intercalary both are involved in vertical growth. Vertical growth. But the lateral, this lateral meristem involved in secondary growth. I will clearly explain in secondary growth. Okay. Remember anyway, apical and intercalary meristem in which growth? Vertical growth, and lateral meristem involved in radial growth in secondary division, secondary development. Right? Is it clear?
Yes, sir.
Position
Yes, sir.
clear?
Yes, sir.
Right.
Yes, sir.
Now, after position, plane of division. In plane of division, first let me complete about rib division, plate division, and mass. And first rib or file meristem. For example, rib division. So how the rib division occurs, the cells rib meristem divide only anticlinally. Anticlinal division means, for example, this is the cell. This is the cell. This division called as anticlinal division. What happens if the anticlinal division takes place? So mainly, this derivatives, the rib meristem, are present long rows. Right. So where is, just wait. What is this here? Rib plate mass. Actually, remember, first of all, plate meristem, second rib meristem, third mass meristem. Mass meristem, remember it. First, let me complete plate. In plate division, what happened? Any given types, bounding surface, right? Surface area without increased number of mesophyll layers. Okay. Anticlinal, right? Right. Actually, this is this one anticlinal division. Anticlinal. In rib, periclinal division. In rib, periclinal division. Right. So just imagine, due to this anticlinal division, surface area increase like this. Surface area increase. Due to the rib division, layers increase. The mass irregular division. The mass is irregular division. Simple, while the formation internal structure, what are the internal parts of the stem? Generally, tell me. Hello? What are the internal parts? Stem or leaf or root? If you take internal, what are the three regions
Cortex,
in order?
cell wall,
Sir,
mesocyst,
Right. Epidermis. Right. You tell me epidermis, how many layers in general?
one layer, sir.
One layer. So due to this anticlinal division by plate meristem, as the surface area increase, only layers, layer surface area increase like that. Single layer, uniseriate layer like that. So it is formed as the epidermis. Now it is formed like epidermis formation. So as it is single layer, uniseriate. Now rib meristem, periclinal division. Now the periclinal division like this. What happened during periclinal divisions? Layer increase. First, second, again periclinal division, third. Now the layers, number of layers increase, not surface area. Apart from the surface area, layers increase. Now what is the next after epidermis?
Hypodermis.
Right, hypodermis. How many layers?
2 to 3 layers.
Next,
Cortex.
cortex,
how many layers? How many layers?
5 to 6 layers.
Right. So these all cortex is this is completely called as cortex region. In the cortex, divided as you said, hypodermis, normal cortex, endodermis. Of course, they are the part of the cortex, but it is not a single layer, not a uniseriate. The multi-layered, formed by with help of rib division. The finally, in some portion, some of the part of the steel also formed with help of the rib division. The final mass division, there is no plane due to irrespective division. The steel region formed, steel part pericycles, mainly medulla region formed, medulla region, vascular bundle, they are all formed within the steel by meristematics, mass cell division. So this is plane of division. So anyway, due to the plane of division, all the internal parts are formed. This plane of division occurs in meristematic cells. Based on the cell, they may be epidermis, may be cortex, may be steel. Right. Is it clear?
Yes, sir.
Right. Next, based on function. Based on function, three types. They are protoderm, procambium, ground meristem. The name itself given protoderm. After division in plate meristem, after anticlinal division, protoderm formed. It is the outermost layer of the hang growing region which is ultimately developed into epidermis. Right? Uniseriate. Next, procambium. Next, procambium. It is composed of elongated prosenkyma meristematic cells which develop into primary vascular tissue. These are derived from the procambium, different into phloem and xylem. Right. Now the procambium cells, they from the laterals, from lateral meristems, procambium cells. Sorry, from procambium, lateral meristem develops. So actually, procambium cells involved in formation outer side phloem, inner side xylem. After formation, the middle, this one. Actually, this is one in one bundle, that is procambium. Let me tell me in detail. For example, how you see this TSF shoot showing? Hello?
Yes, sir. Yes, sir.
Right. So if you take one vascular bundle, cambium cells, cambium cells, cambium cells, cambium cells, they are also cambium cells. All these cambial cells are presented not a central, not a outer. These are lateral side arrangement. That's why this all procambium tissues altogether called as a lateral meristem. Right? This procambium function formation xylem and phloem cells and also involved in radial growth and involved in secondary growth. And next, ground tissue. What is ground tissue? Except procambium. Except procambium, except protoderm, rest of the all part ground tissue. What comes under it is a ground tissue. Hello? If you take internal, what are the internal parts? Already one of the internal epidermis completed, clear. Vascular bundle also completed. What is the next in internal? Hello? If you take internal, what is next? You tell me what are the internal? Someone told earlier. Internal parts of the stem and roots.
Sir, cortex.
Right. Right. In the last from the mass, for ground meristem, cortex and only cortex.
Pericycle.
What, right, pericycle and
Medulla.
medulla. Are you able to see the PPT slide?
Yes, sir.
Clearly visible? Classification meristem.
Yes, sir.
Right. Medulla also ground tissue. And next, except epidermis, except vascular bundle, rest of the all means complete cortex, as you said, pericycle, endodermis, medulla, simple. They are all ground tissue part. They are all ground tissue part. So ground meristem forms all this. So this is the function. So function of protoderm, epidermis, function of procambium, formation vascular bundle, function of ground meristem from formation cortex, pericycle, medulla region. Is it okay?
Yes,
Hello?
sir. Yes, sir. Yes, sir.
This is very, very important and very easy. Okay? Right? Any doubt in this?
No, sir.
Classification, right. So the formation tissue is clear. Formation tissue means from the embryonic cells, how the meristematic developed, how the permanent tissue developed. In this part, first we have completed only meristematic tissue. Okay? Now let's shoot apex theories, vegetative shoot apex theories. They are not an important one, but shoot apex in angiosperms important. Shoot apex theories, angiosperm important. Root apex theories, angiosperms important. But let's discuss angiosperm, pteridophyta, and also gymnosperms also. If you take pteridophyta, the first diagram, the first and second, they both mainly from the pteridophytes. Pteridophyta. Mainly. And next, Gymnosperms. Gymnosperms. First, let's say Pteridophyta. Mostly the pteridophytes most probably apical cells involved in formation of the all tissues. One apical cell, one single cell with the tetrahedral shape or three faces, one, two, and backside three faces, and which is involved in formation of three regions from single cell. Epidermis region formation, cortex region formation, and steel region formation from single cell. And some of the pteridophyta group of cells also involved in this. And also in our books, given some of the examples also, page number 354, 16.5.1, the apical cells and also apical initial. Apical cells or apical initial, these are the initial cells to formation body parts. The tetrahedral apical cells occurs in xylotales. Example, xylotales, ecdysium, and ferns. So anyway, remember one thing: the plant body arises from the single tetrahedral cells and its derivatives. But in some water ferns, the example also given, Azolla salvinia, and in some Salaginella species, the apical cell is lenticular, three-side. If you see the diagram, the three-sided, two cutting faces. So anyway, it is three-side or four-side faces, but single cell, most probably single cell or single cell derivatives. Some investigator opined that in ferns and genera, other genera, single apical cell is pyramidal or tetrahedral, four-sided, but this somewhat controversial whether it is four-sided or three-sided. Xylotals, equidistals, and some fern tetrahedral four faces. From water fern, Salaginella, three faces. But anyway, the ultimate is the entire body developed from the single cell. That is the important. And next one, if you see the shoot apex theories in Gymnosperms, cycus type. This is cycus type. And ginkgo type. First, let's see cycus type. In this type, three meristem zones can be recognized. Surface meristem, rib meristem, center. Rib meristem, flank meristem, lateral. This one. Flank meristem. Already you know the surface meristem, surface layer of the cells divide anticlinal, periclinal, diagonally. Cells are not uniform appearance. These cells of the zones give rise to epidermis and other apical meristematic zones. From as its surface area, it forms the epidermis, and other apical meristematic zones. So in this division, anticlinal division, periclinal division, anticlinal division, and periclinal division takes place. Like this. That next one, rib meristem. It is situated central region. The central region of the apex below the surface layer. These cells, those base of the zones divide anticlinally, same periclinal diagonally. Large content, large vacuole pith develop from this zone. As it is center one, medulla. rib. M for middle. Flank meristem, the cells of the zone smaller than those of rib meristem. Rib meristem larger cell, flank meristem smaller cells when compared with rib. And you can assume what portion of the development from the flank meristem. What is the remaining? Epidermis completed, medulla completed. What is the next one? Simple. What is the remaining internal part? Hello? Hello? Are you there? In cycles type,
Yes, sir.
three zones recognized. What are the three zones in cycles type? Three zones: surface meristem zone,
Rib meristem,
rib
flank
meristem
meristem,
center, flank meristem. The surface meristem mainly involved in formation of
epidermis,
right epidermals and apical meristematic zones. And the surface meristem also both divisions, anticlinal, periclinal division takes place. As rib meristem is situated in middle region, here periclinal, anticlinal both divisions take place and forms only with develops, medulla develop. Middle is the center port of the organ, whether it is root or stem. Now the flank lateral and flank cells zone smaller than those meristematic. These from the flank zone, what are the rest of the remaining? Rest of the region?
cortex,
Epidermis. Right. Epidermis completed and pith completed. Cortex and steel region. Simple. Cortex from this flank region. Here, let me try. Cortex. And steel portion. Steel portion except middle. Steel portion except middle. Remember. Steel portion except middle. Right. Next one, Ginkgo type. In this type, five meristematic zones. First one, surface meristematic. No changes. First of all, surface meristematic like a cycus. Right. Central mother cell, second one. Here. This region. This is surface. This is central meristem. Central mother cell. Central. But draw this. Central mother cell. So in central mother cell, this zone occurs in median position below the surface. Below the surface, we can see in diagram. First surface and below the surface, this one. It consists large nuclei, many vacuole, thick cell walls. This already undergo some further divisions. In this rib meristem, plate meristem, mass meristem. The cambium-like transaction shows cup-shaped present in below. It is located below the central mother cells and rib and flank meristem. The cambium-like zones is not uniform. It forms a transitional zone between the central mother cell zones below them and undergoes frequent cell division. So anyway, let's see one by one the rib division, flank division in this. Where is the rib division? Rib meristem. Where is rib meristem? Always rib meristem portion, central portion. Right? The cells zones arranged in rows and pith of the stem. The like a cycas. Here also rib one formation in medulla. This reason rib medulla. Flank meristem. Ear flank. What forms? What the form from the flank meristem? Same cortex and steel except the medulla region. Is it clear? The only few are the extra part here, the whatever it may be here. The when compared with cycas, surface common, but there is no central mother cell region. But in cycas, but in ginkgo central mother cell region extra present. And in this part, meristematic rib meristem central present, flank meristem also present, either side. So one central, two rib, three flank either side, 4 and 5. 5 regions complete. Right, shoot apex theories in angiosperms. This is from this important. So anyway, the shoot apex theories or root apex theories involved in formation roots and shoot. Right. Here given apical cell theory, estrogen theory, tunica corpus theory. Right. Are you able to see slide?
Yes,
Yes,
sir.
sir.
Right. This is important. Shoot apex theories in angiosperm important. Root apex theories in angiosperm important. Right. First of all, apical cell theory. Already told you from beginning the pteridophytic theory, pteridophyta apical cell theory. This apical cell theory proposed by, let me take pen. This apical cell theory proposed by Hofmeister and supported by Nageli in this single apical cell structural and functional unit. Only one. And remember, single apical cell are from the single apical derivatives also involved. Already told you 4 phases, 3 phases in pteridophyta. Next important theory, estrogen theory. This is important for short answer also. Estrogen theory and tunica corpus theory. Estrogen theory, the proposed by Hornstein, supported by Strasburger. According to estrogen theory, the shoot apex consisting 3 distinct zones. 3 distinct zones are dermatogen, peribulum, plerome. If you see diagrammatically, center plerome, group of cells, center plerome, and next, let's change color. This is peribulum. Periplex, periplex zone. Of last final dermatogen. Let me change color in dermatogen. Dermatogen. Three region. So this is called estrogen. Actually, estrogen group of cells. First of all, group of cells. These are differentiated into plerome, periplex, and dermatogen. Right? Already given from the dermatogen. Dermatogen, it is outermost layer. It forms the epidermis. Completed. Periplex, middle layer. Middle layer. This middle layer region formation gives rise to cortex. The plerome, you can guess the innermost layer gives us a steel and all steel pot. Right. Next one, tunica carpus. First estrogen clear, diagram clear. Hello?
Yes, sir.
Right. Next one, tunica carpus. Tunica carpus. Sorry. Change color. Which is proposed by Smith. Tunica and carpus, two zones found in apical meristem. In group of apical meristem, two zones clearly found. One is tunica, another is carpus. The tunica peripheral zone of shoot apex thus forms the epidermis. Carpus inner zone shoot apex that forms the cortex and steel, both cortex and steel. Take diagrammatical representation, Smith. Only two regions, carpus. This is tunica. Green color tunica, center inner one, carpus. So from cortex, some cortex, cortex and steel formation, both cortex and steel, both formation form from the this region. Epidermis from the tunica region. Right? So this theory, most of the scientists act for angiosperms. This is most of the accepted the tunica carpus theory. Most of the scientists accepted. Right. Next, what is a new initial knowledge? Cytohistical generation knowledge. Right. Next one. Types of vegetators: shoot apex, monoplex, simplex, duplex. These are very easy. If you see diagram, they are easily understand. Page number 361. Page number 3. Hello, are you there?
Yes, sir.
Page number 36. See the diagram 16.9 given. They are the types of vegetative shoot apex. They are not important, but very easy. See once again. Monoplex, simplex, duplex. If you see monoplex diagram, how many initial cells are there? Mostly in diagram first monoplex diagram. Monoplex diagram only the one cell, one initial, one are these derivatives. A simplex type, if you take the simplex type, only uniseriate cells, group of uniseriate cells like this. So division takes place in this layer, this group of cells. In duplex, two layers. So cell division takes place in this group of cells like this. Very simple. And root apex theories. What are root apex theories? The same like a shoot apex, root apex theory is also there. If you see root apex theory, again apical cell theory common. And estrogen theory common. Same estrogens applicable for both shoot and root. Apical cell theory applicable for both shoot and root. And next another one is the Carper-Kappa theory. Shoot Carper-Kappa theory. Right. So these are the shoot apex theories diagrams. Estrogen theory. So apical cell theory, estrogen theory, tunica corpus theory. See the diagram. Plerome region, peripheral region, dermatogen. Three region clearly. This group of cells called as estrogen. Already told you. The group of cells in this, just wait. This group of cells, estrogen. In this in tunica corpus, in group two regions, corpus central, tunica cap. Now root apex theories, common apical cell theory, no need to explain, and also estrogen theory, but somewhat different in diagram. Here also same group of divisions, group of cells in here, but different in the root apex. This is from the root apex. According to root apex estrogen theory, Hornstein, Starberger, four region. In root, actually, in shoot, three regions there, but in root, four regions. What are the four? Dermatogen, like a shoot apex, common. Peripleum also common. Plerome, these three common. Calyptrogen, new one. Calyptrogen. As we discussed in shoot apex theory, dermatogen, epidermis, peripleum, cortex, plerome, steel. The what last one, it gives rise to root cap. Only one. Root cap is one. So this is root apex theories. And also some of the in book here some vascular cryptogams, gymnosperms, dicotyledons. Also given ranunculus type, causalana type, common type. If you see ranunculus type, page number 364, page number 364. In ranunculus type, all the regions of the root, including root cap, develop from the single row. In some plants, this is only estrogen. So other formations, other cellular configuration. If you see other cellular configuration, the root apical meristem divided into different type. One is ranunculus type. In ranunculus type, in root apex, or only single row of cells. From the single row of cells, all four regions develop. What are the four regions? What are the four regions? In root. Hello?
Dermatogen,
Right,
Cerebellum.
dermatogen, peripleum, plerome, and colprogen, right. And another one is Casuarina type. A Casuarina type is mainly found in the Gymnosperms. There are two types, root apex, one row, second row. In this type, inner row of initial forming the inner cortex and central cylinder. This one. Outer row forming outer cortex and root cap. Simple. Outer cortex and root cap. And epiblema. Dermatogen plays epiblema arises from the outermost layer. Common type. The common type is present in dicots. In this, the three rows present. One, two. And in monocot, four rows. This is dicot. One, two, three, four. So anyway, these are the development of root. Right, another development of the root pods, Carper-Capet theory. Carper-Capet theory. If you see the Carper-Capet, right, Carper-Capet theory, which is proposed by Shaw. There are two regions. What are the two regions? Hello?
Carper and Cappe.
Right, Carper and Capet. The Carper forms the body and Capet zones form the cap. Right. Simple. Body parts. Epidermis, steel, cortex. These all parts is epidermis region, blue color, steel region, cortex region, either side. Carper. This is from Carper. And Capet zones forms the air cap. And this is equivalent to the tunica carpus theory and shoot apex. In tunica carpus also, how many zones? To
Two,
formation shoot?
two
Two.
zones.
Right, here also two zones, Carper and Capet. And the two divisions are distinguished by the type of T, also wide division. Carper is characterized by inverted T, Carper, Carper, Carper region characterized by inverted T division and Capet by T region. Simple. If you see Carper or T, where it is by the type of T also called Y division. Okay. Carper characterized by inverted T. So here. T or Y shape like this. So these cells present in Carper and Capet state T. Capet region. Capet region. Like this. Carper division. This one. And on a mantle core there or not? Carper Cape, mantle core not given. Okay. Okay. D division and D division example the possible in GMS Poaceae family given. Right. Concept of quiescent center. Quiescent center. A British scientist Close proposed by this theory. Where is marker? Proposed this theory to explain the root apical meristome activity. These centers located between the root cap and differentiated cells of the roots here. This is root cap. This is the differentiation portion of cortex and steel and cortex. So this is the center. The apparently inactive region of the cells root from promeristome is called quiescent. Inactive region. This quiescent is meaning inactive region. And it is the site of hormone synthesis and also the ultimate source of the all meristematic cells of the meristome. So the meristematic production from the quiescent region, which is present in the center, the cap and differentiation region, and which is was discovered in GMS. British scientist GMS discovered by Close while making autoradiography studies DNA synthesis in root apex GMS. He discovered the central cup-like region inactive cells in this root apex and the name given as a quiescent. The cells of the region have the lower concentration of DNA, RNA, protein, fewer mitochondria, where the mitochondria high in number, mitochondria majorly found in where the active cells are present like meristematic, there high rich abundant mitochondria present. But is it an inactive state? The all are less content, DNA less, RNA less, and protein less, very less few of mitochondria, and little endoplasmic reticulum, very small diffusomes, all are very, very less. These cells divide very rarely. Right, the quiescent center found a large number of plants. The main is studied by B.D. Deshpande in Amaryllidaceae. Generally, the cells of quiescent center do not divide, but they become active when previously active cells get damaged. Right. Thus, this centro acts the reservoir of the cells as permanent source of active cells. Because actually, what happened here, the cells quiescent center do not divide, but they become active when previously active cells get damaged. When active cells damaged, they become active. If already existing previous cells if damaged, they become active. Thus, this is act as a reservoir of the permanent source of the active cells. Of course, they are inactive state, but any damages occur, they can active again. And Close has also shown that most of the roots exposed to toxic doses, X-rays, the meristematic cells stop the synthesizing DNA. The cell's quiescent center become meristematic and start synthesizing DNA. So I already told you just now, when any damages occur, any damages means if malformation in DNA cells, the quiescent center become meristematic and synthesizing DNA. This is because the cell's quiescent center are more resistant in irradiation than actively dividing cells. But if you take the active dividing cells, any malformation, any untoward actions occur, they cannot divide. But what happens quiescent center, if any untoward actions occur in the cells, automatically it can change into meristematic cells and all retain the all character of meristematic cells and actively divided. According to Close, the cell's quiescent center are inactive because their position in the apical meristem and not due to any inherent ability to undergo division. Right, this is about quiescent center. Quiescent center also important for short answer. This is very, very important for short answers. Like, clear? Hello?
Yes, sir.
Right. Now, meristematics, types, shoot effects, root effects theory completed. Next one. Tissues and tissue system. Are you able to see the slide? Types of plant tissue, meristematic, apical, lateral, all completed. Hello?
Yes, sir.
Yes, sir.
Right. Meristematic tissue completed, types of tissue completed. Now permanent tissue. In permanent tissue, first let me complete about simple. In simple tissue, parenchyma. Already just 1 week back, we knew about this parenchyma, collenchyma, sclerenchyma during practical training classes. Tell me about parenchyma. Please cooperate with these simple topics. Then only we complete all the topics, all complex topics. Please cooperate. Tell me, parenchyma, what you learned during practical training class?
Unspecialized tissue, sir.
The very, very common, actually the parenchyma,
Living tissue.
right? Okay, as the definition, the simple tissue, the all type of similar cells are there that function and structural similar, right? Let's see the structure of the parenchyma. Before that, the parenchyma is the fundamental basic tissue and which is the primitive tissue. And if we see the parenchyma cells, the parenchyma cells with intercellular, without intercellular space. Hello? Arrangement.
With intracellular.
Yes,
With intercellular space and the shape of the parenchyma cells may be global, oval, or the various forms of the shapes. And tell me the cell structure, a parenchyma cell structure in the tissue. If it takes one parenchyma cell, which is covered by cell wall or not, cell wall present or absent? Hello?
Present, sir.
Right, cell
present,
wall
sir.
present. What is the cell wall component? Chemical composition?
Cellulose, sir.
Cellulose. Right. So mainly the parenchyma mainly found in softer regions of leaves and stems and roots and fruits, right?
Yes, sir.
The parenchyma is diverse, apical, and also origin of parenchyma is diverse. The apical meristem produced in stem and root, and the marginal meristem in leaf. Parenchyma of the xylem and phloem develop from the vascular cambium, and that of the secondary cortex from the cambium. So these are the origins, and this is mainly isodiametric shape. So anyway, the shapes also different, but types of parenchyma. Remember, when any soft tissue parenchyma present. Types of parenchyma. Where are the parenchyma cells present inside the plant?
Roots, stem,
Right, root, stem, leaf. For example, if you take the leaf, chlorenchyma cells are present. Chlorenchyma. Chlorenchyma means if you take the leaf internal structure,
Leaves.
right, epidermis, lower epidermis, upper and lower. What is the rest of the position after the upper and lower? Spongy tissue, palisade and spongy tissue, mesophyll cells, right? Mesophyll cells, mesophyll cells, right? These mesophyll cells, one type of parenchyma, and this is called as chlorenchyma because which consist chlorophyll pigment. As it consists chlorophyll pigment, this type of parenchyma called as chlorenchyma. And another important one is aerenchyma. In the name itself given aerenchyma, what is present inside aerenchyma? Another type of parenchyma tissue,
Parenchyma.
Yeah.
which is I would like to give the simple clue, which is
Air,
mainly found in water, right?
air, sir.
Air, mainly found in water plant. And another storage part of food parenchyma, storage food parenchyma, and
Storage.
also water food parenchyma. Any storage region, food storage region, this type of parenchyma present, which is storage sugar and other carbohydrates. Can you give the example? Can you give the storage parenchyma example part?
Tubers.
Huh?
Tubers.
Tubers, sir.
Tubers, simple tubers. Like that,
Potato.
water storage parenchyma. Right, potato. Water storage parenchyma. Another type. The name itself given. What storage in this parenchyma cells?
Nutrients.
Water storage parenchyma. Water. Example.
Hydrophytes.
No, actually, water storage parenchyma not in hydrophytes, in xerophytes.
Yeah,
Again,
yeah.
Opensia. If the best example,
Aloe
if
vera.
you right, aloe vera the best example. Right. Another type of parenchyma, idioblast. In idioblast, which is produced and storage of accumulate tannins and calcium oxalate, and also store myosin, oils, mucilage,
Seeds.
resins. Right. Mainly they are present in seeds, and also bark, stem also, roots also present, but majority in seeds. Right. This is short answer. Parenchyma short answer. Remember. And next one, colon. When compare with coloncyma, coloncyma, parenchyma difference. Hello.
Yeah. Coloncyma. The corner region much thickened.
Not only corner. When compare with the in general, when compare
Yeah.
with the parenchyma, coloncyma cells some more thicker.
Yes, sir. More thick.
Because actually the pectin substances present in the cell wall. In primary cell wall made up of cellulose, pectin is also present.
To reduce the pressure, high
And
pressure
also it gives the
branches.
mechanical strength to the body.
Yes, sir.
And based on the composition of the pectin substances, there are different type: angular coloncyma, locular, plate, annular, 4 types of there. Clearly given in your book also. A diagram, B and C. The based on diagram you can observe. And tomorrow we will complete about sclerenchyma, sclerites and fiber, xylem and phloem. All. Any doubt today class?
No, sir.
Hello? Right. Can I quit?
Yes, sir.
Thank you, one and all.